Hi everyone! So the purpose of the research paper I looked into was to study potential treatments for Alzheimer's disease through the use of mesenchymal stem cells, or MSCs, in mice. Alzheimer's is a slow, progressive, detrimental degeneration of the central nervous system seen through inflammation, synaptic and neuronal dysfunction, and overall deterioration. Neuroinflammation is primarily orchestrated by microglia, which become overly active and end up harming neurons and progressing the development of Alzheimer's. So this study is looking into targeting neuroinflammation as a potential treatment. Mesenchymal stem cells were chosen as they are known to interact with cells in the immune system by supporting growth and having immunoregulatory properties. They release extracellular vesicles, or EVs, which lessen the activation of the harmful microglia, causing inflammation and loss of dendritic spines, which are the branches reaching out on dendrites that help to receive information from axons. Research involving the extracellular vesicles of mesenchymal stem cells has been done before, but the unique aspect of this study is that they're administering the treatment intranasally, or through the nose of the mouse. This method is both safe and low invasive, which is promising for the potential translation of this treatment from mice to humans. The study also used 3XTG mice, which are mice that express 3 mutant human transgenes that are associated with Alzheimer's disease. There were 2 groups of mice, an experimental group given the extracellular vesicles suspended in PBS, and a control group given just the PBS vesicle with no extracellular vesicles. In order to isolate the extracellular vesicles from the stem cells for treatment, a process of repeated centrifugation was performed that acted to remove cells, cell debris, and other contaminants other than the extracellular vesicles. The mice were given 100 microliters of their respective treatments 2 different times, and 21 days later were processed for microglia activation and dendritic spine density. So immunofluorescence was used to investigate microglia activation in different regions of the brain, and cell density and cell soma size were used to quantify this data for comparison. The figure on the left here shows the images of microglia, which are in green, in the hippocampus, entorhinal cortex, and prefrontal cortex, respectively. The top row shows images of the control mice, and the bottom row is mice treated with extracellular vesicles. There is a clear difference in the 2 rows of images, with the animals receiving EVs having a reduced number of microglia compared to the control. The graphs below show that the differences in quantifiable data gathered from the immunofluorescent images between the groups are statistically significant. Further, the damaging or reactive phenotype of microglia can be identified through their larger soma size, and the EV treatment caused a significant decrease of microglial cell soma size. The figure on the right shows a Golgi stain of dendritic segments from the same parts of the brain where microglia activation was studied. Dendritic spine density was compared in the 2 groups and was determined using the number of dendritic spines and their lengths. The EVs are shown to increase dendritic spine density in 3XTG mice, with the treatment group having a significantly higher number of dendritic spines compared to the control group. So, this has relevant value as a possible therapy because limiting the expansion of these harmful microglial cells has beneficial consequences in mice with Alzheimer's. The MSC EVs act to reduce the presence of the overly activated microglia, which result in less harm to the neurons, which can be seen in the Golgi stain showing higher dendritic spine density. The presence of more dendrites reduces synaptic dysfunction and allows for better communication between neurons. Previous studies that have administered this MSC-EV treatment yielded similar beneficial results, but were done intravenously or intracerebroventricularly, so directly into the vein or the brain, and were also done over the course of weeks or months. The treatments administered in this study were done through the nose and consist of only 2 injections. So this safe, low-invasive, and effective method is super promising for future use in humans with Alzheimer's. In the future, research can be done to possibly increase dosage of this treatment and determine if that could lead to potentially a greater positive impact on the patients. So let me know if you have any questions. Thanks for watching.